5.3 RNA World. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution, but briefly we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc., evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA become double-stranded and having complementary strand further resist changes by evolving a process of repair. 5.4 Replication. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication, figure 5.6. 5.4.1 The experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Michelson and Franklin Stahl performed the following experiment in 1958. Number 1. They grew E. coli in a medium containing N15H4Cl. N15 is the heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride CsCl density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N14 only based on densities. Number 2. Then they transferred the cells into a medium with normal N14H4Cl and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on cesium chloride CsCl gradients to measure the densities of DNA, figure 5.7. Can you recall what centrifugal force is and think why a molecule with higher mass or density would sediment faster? Number 3. Thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, second generation, was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid densities DNA molecule? Very similar experiments involving use of radioactive thymidine to detect a distribution of newly synthesized DNA in the chromosome was performed on Vicia faba beans by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicate semi-conservatively. 5.4.2 The machinery and the enzymes. In living cells such as E. coli, the process of replication requires a set of catalysts that is known as enzymes. The main enzyme is referred to as a DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 6 base pair, compare it with a human whose diploid content is 6.6 into 10 to the power 9 base pair, completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with a high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically replication is a very expensive process. Deoxyribonucleoside triphosphates serve dual purposes. In addition to acting as substrates, they provide energy for polymerization reaction, that is, the two terminal phosphates in a deoxynucleoside triphosphates are high energy phosphates, same as in case of ATP. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to the very high energy requirement, the replication occurs within a small opening of the DNA helix referred to as replication fork. The DNA dependent DNA polymerases catalyze polymerization only in one direction, that is 5 to 3. This creates some additional complications at the replication fork. Consequently, on one strand, that is the template with polarity 3 to 5, the replication is continuous, while on the other, the template with polarity 5 to 3, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase, figure 5.8. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite reason in E. coli DNA where the replication originates. Such reasons are termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, a chromosomal anomaly. You will learn the detailed nature of origin and the processes occurring at this site in higher classes. 5.5 Transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now form base pair with uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated, in transcription only a segment of DNA and only one of the strand is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Why both the strands are not copied during transcription has the simple answer. First, if both strands act as a template, they would code for RNA molecule with different sequences. Remember, complementarity does not mean identical. And in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, should be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. 5.5.1 Transcription Unit. A transcription unit in DNA is defined primarily by the three regions in the DNA. Number 1, a promoter. Number 2, the structural gene. Number 3, a terminator. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is from 5 to 3, the strand that has the polarity 3 to 5 acts as a template and is also referred to as template strand. The other strand, which has the polarity 5 to 3 and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription. Strangely, this strand, which does not code for anything, is referred to as coding strand. All the reference point while defining a transcription unit is made with coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. Can you now write the sequence of RNA transcribed from the above DNA? The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5' end upstream of the structural gene. The reference is made with respect to the polarity of coding strand. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strands. By switching its position with terminator, the definition of coding and template strands could be reversed. The terminator is located towards a 3' end downstream of the coding strand, and it usually defines the end of the process of transcription. Figure 5.9. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. 5.5.2 Transcription unit and the gene. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural gene have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. 5.5.3 Types of RNA and the process of transcription. In bacteria, there are three major types of RNAs: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings amino acids and reads the genetic code, and rRNAs play structural and catalytic role during translation. There is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription, which is known as initiation. It uses nucleoside triphosphates as substrate and polymerizes in a template dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continues elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off, so also the RNA polymerase. This results in termination of transcription. An intriguing question is that how is the RNA polymerase is able to catalyze all the three steps, which are initiation, elongation and termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor sigma and termination factor rho to initiate and terminate the transcription respectively. Association with these factors alter the specificity of RNA polymerase to either initiate or terminate figure 5.10. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, that is, there is no separation of cytosol and nucleus in bacteria, many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. Number one, there are at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. The RNA polymerase 1 transcribes rRNAs 28S, 18S and 5.8S, whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5sRNA and snRNAs, small nuclear RNAs. The RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, that is hnRNA. Number two, the second complexity is that the primary transcripts contain both the exon and the introns and are non-functional. Hence, it is subjected to a process called splicing where the introns are removed and exons are joined in a definite order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide known as methyl guanosine triphosphate is added to the 5' end of the hnRNA. In tailing, adenylate residues 200 to 300 are added at 3' end in a template independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation, figure 5.11. The significance of such complexities is now beginning to be understood. The split gene arrangements represents probably an ancient feature of the genome. The presence of introns is a reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. 5.6 Genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences, though, to support the notion that change in nucleic acid genetic material were responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code were most challenging. In a very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, and geneticists. It was George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because a permutation combination of 4 to the power 3, that is 4 into 4 into 4, would generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Har Gobind Khorana was instrumental in synthesizing RNA molecules with a defined combination of bases, that is homopolymers and copolymers. Marcel Nirenberg's cell-free system for protein synthesis finally helped the code to deciphered. Severa Ochoa enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template independent manner, enzymatic synthesis of RNA. Finally, a checkerboard for a genetic code was prepared, which is given in table 5.1. Table 5.1, the codons for the various amino acids. The salient features of genetic code are as follows. Number 1, the codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acid, hence they function as stop codons. Number 2, some amino acids are coded by more than one codon, hence the code is degenerate. Number 3, the codon is read in mRNA in a contiguous fashion. There are no punctuations. Number 4, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine PHE. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. Number 5, AUG has dual functions. It codes for methionine MET and it also acts as initiator codon. Number 6, UAA, UAG, UGA are stop terminator codons. If following is the sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it, take help of the checkerboard. Now try the opposite. Following is the sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. Do you face any difficulty in predicting the opposite? Can you now correlate with two properties of genetic code you have learned? 5.6.1 Mutations and Genetic Code. The relationship between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 4. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutation will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the chain of amino acid residue glutamate to valine. It results into a diseased condition called as sickle cell anemia. Effect of point mutation that inserts or deletes a base in structural gene can be better understood by following simple example. The conclusion from the above exercise is very obvious. Insertion or deletion of one or two bases change the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutations. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards.